Okay. If the last few remaining standing people could find a seat, then we can try to get going on time this afternoon. Thank you very much. And welcome to this symposium, which we start off with this afternoon and we continue tomorrow morning. Organized by the Biodiversity Genomics Europe project, and talking a little bit about the project itself and where we've come to up until this point, but also inviting a lot of our friends and colleagues working in the field of biodiversity genomics to really start to think about where we are today, and where we want to be tomorrow, and how we're going to get into a future of a real biodiversity-enabled system in Europe. And before we set off with this first session this afternoon, we have a few welcome remarks from our host here at the Institute, Michel van Kamp.
Okay, thank you. Thank you for coming, numerous colleagues. Welcome at the Royal Belgian Institute of Natural Sciences. So just first a quick word about our wonderful institute. It's not only a famous museum with iguanodons, it's also a top scientific institute with about 170 scientists bringing research right where society needs it. And in particular, we provide a lot of advices to politicians and the citizens. The theme today is unlocking the power of genomic science for nature and the bioeconomy in Europe. Is exactly what we are doing, linking cutting-edge science to the big sustainability challenges we face. To protect our natural resources properly, we need much better biodiversity data and lots of it. We can find a lot here and all together, and thanks to genomics, that's now possible, and in particular, DNA-barcoding and an environmental DNA let us detect species fast and cheaply. The whole genome sequencing helps us understand the health and diversity of populations. And there are quite a lot of fascinating results with that. These tools do not replace the usual surveys they add to them. They help us protect habitats, monitor water quality, check fish stocks, and even improve transparency in farming. And this all supports important European policies like the Nature Restoration Regulation. And I hope it will last because there are other priorities today. And our global biodiversity commitments. Europe has big strengths, and we forget it too much. World-class laboratories, expert scientists, some of them, all of you, here today, active networks and strong standards for open and reliable data. And that's a big challenge as well. But to really make a difference, we need to coordinate better across countries, build complete DNA reference libraries, make data easy to share, and bring all countries up to speed in skills and tools. This symposium is all about that. It's a chance for scientists, policymakers, environmental groups, and business to come together and plan how we build a Europe-wide system for biodiversity and genomics. A system that first helps nature and supports a sustainable bioeconomy. I am looking forward to your discussion over the next 2 days, and I'm sure my colleague will inform me, for instance, Anna, and to seeing how we can make this scientist promise real for Europe and our planet. And Last but not least, please do not forget to have a look at our wonderful galleries. So I wish you a nice meeting.
Thank you very much, Michel. I realize I forgot to introduce myself at the beginning of this session. My name is Rob Waterhouse. I'm sitting at the SIB, Swiss Institute of Bioinformatics, and part of the coordination team of the Biodiversity Genomics Europe project. So in this first session, we will hear from the communities, both the global level and the European level, the communities of barcoding and reference genomes. And I think in the interest of time, we will proceed through each of the different presentations. And there is a half an hour slot at the end for discussions. So I encourage you to note down your questions so that we have a fruitful discussion after the presentations from the communities. But before we go into that, we'll have introductory remarks from Dimitris Koureas, our director.
Thank you, Rob. Good evening, everyone. Michel, thank you. Where's Michel? Thank you very much for your comments, and I think thank you for hosting us. I think you've set the scene. There's not much for me to add, apart from saying that sometimes we forget that we are operating in the space of science at the same time navigating a huge transformation of our societies. A transformation of our societies that to a certain extent is based on the availability of more information than we ever thought possible to be able to deliver. Big data is not just changing the way we understand science, it's changing the way we perceive the scientific practice altogether. And I think this is the landscape in which we have to move forward. And biodiversity genomics is not exception. Every 2 years, the INSDDC repositories, that they are doubling their holdings in data. 75% of all the data ever deposited in these repositories were deposited essentially in the last 4 years. That shows a lot how technology is really helping us scale up exponentially the amount of information that we generate. What do we do with that information? How do we interpret it? Riding the wave of big data is much more difficult than it sounds. At the same time, the technologies that allow us to generate all of this information are becoming available to everyone. Every single lab, every single individual PhD student can generate and be part of that community of data generation. And it's important that that happens, because most of the questions that we have to answer when it comes down to biodiversity, they have to be addressed at the local scale. So it's important that that democratization of the technology access and data access happens where it is most needed, and that is the local scale. At the same time, there are big issues we have to all address, continental or global challenges that we face, whether they derive from the policy frameworks or from the very need of sustaining our ecosystem services. Moving from that local scale to the global effort is something that I hope we will be able to initiate, at least at the European level. We have all of these amazing projects taking place at national level, that they can work together, they can join forces. And we have our North Stars, big initiatives like iBOL or EBP. We have all the components in place. We have the European networks, iBOL Europe, ERGA, CETAF, that really can help us organize our work. The space of opportunity is really growing, but the time we have to embark on it is closing. So it is, hopefully, I hope from today and tomorrow, we will have the opportunity to start thinking together what are the next steps to be able to make that local democratization of technology and data generation something more than the sum of its parts. And in that process, I hope that policy stakeholders, also the European Commission, will be on our side, on the side of science. We have seen these examples, and I hope that we will continue seeing that support over the next years. That's my few words. I hope we have an amazing 2.5 days, and I also hope that you are able to enjoy Brussels to the extent that you can. With that, I would like also to introduce Giles Doignon. Giles is a policy officer. He's leading the biodiversity team at the DG RTD. And very recently, I think a few days ago, I'm very happy to say that he was appointed the European Union focal point, primary focal point for IPBES. So I think he's the right person to give us some introductory comments about the role of the Commission in this. Thank you.
Yeah. No slide. Thank you very much, Dimitris. And there are days where I really love my job. This is one of these days because I am with friends, so Dimitris, Anna from CETAF, Michel from the museum, and many others. And also it's a day where I will learn a lot. So even if I used to be a taxonomist, I'm just discovering genomics with you, and it's just fantastic. So I'm a very old traditional taxonomist. I remember when I had to check some slides from specimen collected in 1890 in Japan. But I also did some analysis with RAPD. I don't know if you know that, okay? Just some general remarks. Well, first, Michel, the Nature Restoration Regulation is a regulation, so we have to implement it. That's very important. And it's also particularly important when we see the state of nature in Europe with the last report of the European Environment Agency yesterday. And it's not good. And also, last week was a report about the 7th planetary boundary being reached, so ocean acidification. But when you look at all the planetary boundaries, the worst one is genetic diversity. So, as Dimitris said, I have the honor to be appointed IPBES EU focal point. So I think it's very important to take a distance and keep in mind the big picture. So IPBES, in its first global assessment on biodiversity in 2019, had one key message: transformative change. So we need to change the way we produce, the way we consume, the way we look and respect nature. Because if we continue, biodiversity loss will continue and we will be doomed. And the key message is also that we have to protect, to restore, but also to sustainably use biodiversity. So that's why I would put the needs for biodiversity and the sustainable bioeconomy. But we also need to change the way we learn about biodiversity, how we understand it. And this is where genomics are just a fantastic new frontier for science. Just imagine if Darwin would have known, he would be freaking out, all the fantastic things we can do with genomics. And I will remind one sentence from my professor of zoology at the University of Brussels saying, "It is impossible to know something you do not know." And actually, thanks to genomics, we know much more. So I will not go into all the details, you are the expert, but with the use of barcoding, eDNA, eRNA, there are so many new species or group of organisms. So we know more about the species on this planet, but we also know more about what we are losing, what we are destroying. Just think about all the species, the new species in the soil. We know more about their interactions, the complexity of biodiversity, and we have also new tools, thanks to genomics, to monitor, to do spatial planning. So for us, and yes, Dimitris, at least from DG Research, you know that we will support you. Science is the basis. Knowledge is the basis. So taxonomy, genomics, this is the basis. And so if we want to develop a so-called nature-positive economy, society, and that's what we should do, we need to know. And this is why the European Commission already supported and funded many research projects on traditional taxonomy, such as Tetris, on genomics, Biodiversity Genomics Europe, and we will continue. I presume you all know about the topics under the work program 2025. The deadline was 2 weeks ago. Too late if you didn't know. But there are many other opportunities coming. So I will not go into all the details about the work program 26-27, but there will be also space for taxonomy and genomics in some topics. We also supported infrastructure, such as DISCO. And also now we are really looking at the exchange of data. And these data are crucial for the new life science strategy. And we have our own action in my team, so ready to consolidate the knowledge and the data. You have also the new EU infrastructure strategy, and we are in contact with our colleagues. And there will be also the Biotech Act, and this is where we need data, but we really have to ensure sustainable use of biodiversity. Coming back to pure taxonomy, we need a new generation of taxonomists, genomists, and actually they are there. So many people were telling me when I was a taxonomist at 25 years old that, you know, this is the past, you will finish in one museum, etc. Well, actually, yes, I finished in a museum, and I'm very proud of it. But with Tetris, for example, when you did your cascading grant, you got 53 applications, and that was a very, very nice surprise. So there are young taxonomists, and of course, this generation work on both traditional and genomics. Just also to mention in terms of data that we really want to make the best use of data coming from citizen science. So that's why we had also one specific topic in the work program 2025. And we want all these data to be used. So for example, by GBIF. There are also new opportunities. There are developments in other science, like environmental observation, satellite and remote data. In February, we co-organized the Biospace 25 conference with the European Space Agency. You can look for it. And it was just fantastic because it was the community of the space people, the geeks, and the in situ experts, the nerds. And it was really like speed dating with so many potential collaboration. And that's why we are coming very soon with a topic about the interoperability between in situ and remote data, including genomics. So ready to know about biodiversity from genes to space. Another development is modeling. We know that modeling is already very complex when we speak about climate, but for biodiversity it's even more complex, and you probably know the Nexus assessment by IPBES saying that we have to analyze biodiversity, food, water, health and climate together. And also, almost there, thanks to genomics, you have also other applications, and maybe you do not think about them. It's to help into the deployment of new areas, such as nature-based solutions, and you can help by advising which species will survive in the future in a changing planet. And the last big opportunity for all of us is the use of artificial intelligence, not necessarily to use ChatGPT to prepare a proposal, but really to boost the analysis of all the data. So I will finish here. I'm just saying that I'm really here to learn, and I'm very happy to learn with friends. So thank you.
Thank you very much, Gilles and Dimitris, for, I've gone backwards, for the European perspective of where we are with biodiversity genomics. And now we're going to start off with the perspective from the barcoding community, and we invite Paul Hebert to tell us all about iBOL.
All about iBOL in 10 minutes. Wow. Okay, let's go. So going to advance here. Is that forward? That's the green. Okay. 18 years ago, we had this idea that we would try and launch an international consortium to advance the application of DNA-based identification systems. And it took us 3 years to get things going. In 2010, just about 15 years ago, iBOL launched, and these are the nations that are currently involved in helping to advance its mission. What's it doing? It has 3 grand goals, and here they are. Register every multicellular species. Document their interactions, and then finally establish a global biomonitoring system. How are we going to do it? Through 3 research programs. This was the sort of ambitious research program that was set up in 2010. Things rarely unfold as you think they might, but they're unfolding. Barcode 500K was a 5-year program, launched in 2010, finished on time and budget in 2015. We planned that, you got to sit back and spend a little bit of time deciding about how you're going to push things forward and rally the funds. So we spent 3 years on that and launched in 2019 in Trondheim Bioscan. It's a decadal project. It's in progress now. And when it finished, plans were to spend 2 or 3 years and then launch Biodiversity Science's first mega science project, a $1 billion program, planetary biodiversity, that'll finish those 3 missions that I mentioned in my second slide. Okay, Bioscan. We finished Barcode 500K, so I'm not going to dwell on it, but I will talk very briefly about Bioscan and what its goals were. We wanted to advance species discovery because we all know there's a massive numbers of species which don't have Linnaean names. And we said we'd do that with focused efforts in 3 parts of the world. Europe was one of those, and analyzed 10 million specimens over the decade. We said we would use DNA-based methods to advance our understanding of species interactions, because if you think about it, species are the fundamental particles of biological systems. Chemistry, we've got our table of elements, and chemistry is that business of looking at their interactions. Well, biodiversity science hasn't really begun that. Our grand goal has been to register the species. No, we need to understand their interactions. And then finally, we need to have a detailed understanding, Giles, just picking up on your point, we need to understand where life is distributed on the planet and how it varies. If we're going to achieve the goals of the 30 by 30 that the UN has established, we need to understand where life is present and diversity across the planet. So we planned on analyzing 100,000 bulk samples from around the planet. Those were the goals. In order to achieve those goals, we had to crank up analytical capacity. And here you can see the rise on the left on single specimen analysis, the number of individual specimens that have DNA extracted from them. And it's now about 3 million specimens per year, and those extracts fuel both the species interaction work and the species discovery work. And then the other line of analysis are bulk samples, which are rising currently to about 20,000 bulk samples per year, so thousands of specimens using metabarcoding to infer the species composition. So we've cranked up production. And the impact of this, and Giles, you mentioned your background in traditional taxonomy, it's amazing what's been accomplished over the 275 years, but it's registered species at about 7,500 species per year. And you can compare that with the pace of registration of species at present with DNA-based methods, which are sitting 20 times higher. 150,000 species equivalents per year is the current figure, and there's every possibility to increase that number in a radical way. Of course, they don't have Linnaean names. They're assigned to barcode index numbers, and here you can see one of the 1.3 million barcode index numbers and the data that's associated with it. And sometime next year, the number of species with a barcode registration will exceed the Linnaean total for the animal kingdom. And, you know, we're gathering information, registering species, and it's changing our view of the understanding of life on the planet. And this is one of those changes. For an awful long time, it was thought that beetles were the most diverse group of insect life on our planet, but that's clearly incorrect. It's hymenopterans, it's dipterans, and in fact, this single family of flies, the gall midges, have more species than all of the 185 or so species of coleopterans. So that's one example of which we're changing patterns of species diversity. We're understanding more about species richness. For a long time, it was thought there might be about 6 million species of insects. Now, based on the current progress in DNA-based analysis, we're estimating it's closer to 17 million. Okay, that's all I'm going to say about species discovery. It's progressing. We're learning things. The work on species interactions involves the extraction of DNA from a single individual, like a monarch butterfly caterpillar that you see there, and then amplify a number of different genes to extract information about the host plant of that insect or the parasites, the parasitoids that attack it. Given the brevity of my conversation with you, I'm not going to be able to say too much about this approach, but I can say that we're learning things. Just very quickly dive into two cases of host parasite and host parasitoid interactions. In this case, the darkest of the dark phyla, and I'm so happy Mark is here because he's fascinated by these organisms. I have no idea why, nematodes. But Cotesia is a genus of braconid wasp. So these are the kind of things that we can learn about by using multiple gene markers to probe a source organism and the DNA sequences that you can extract from it. And if you analyze a fair number of insects as we're doing total body extracts, what you find is that in a number of cases you pull nematode sequences out of the insect. And in fact, we have 40,000 insects that have delivered nematode sequences, and those nematode sequences represent 7,000 different species of nematodes, many of them highly specialized in terms of their interaction with their insect host. And it's clear well, we can't estimate how many species of nematodes are infecting insects with this little data. It may well be hundreds of thousands of species, but we know it's certainly way more than 15,000, which, Mark, I think maybe we've got 20,000 nematodes with Linnaean names, perhaps. So there's an awful lot of insects that are inflicted. And look at the nematodes inside that body of that secoded fly. It's been turned into a flying machine that's distributing the nematodes. Quite neat. This is the genus Cotesia. It's a single braconid genus, and we have barcoded 688 species of Cotesia from around the planet. Pink is 81, Costa Rica. Dan Jansen has spent 45 years of his life collecting caterpillars. He and his big team have been collecting caterpillars, have spent $40 million building, connecting those Cotesia with their caterpillar hosts. Fabulous work, and making it possible for me to show you this particular figure that shows for this one group of 41 Cotesia species that are in a single species complex, the range of hosts that they use. Each of them is tightly host specific. There are a few gray boxes. Those are species that we've only collected in a Malaise trap. They've never been reared. This kind of study that was carried out in the ACG in Costa Rica, I'm quite certain will never be conducted anywhere else on the planet. It's simply too expensive. We can extract this information by simply gathering up caterpillars and pulling out the secondary sequences and deriving those connections. And that's in the process of what we're doing now. This is the future. It looks complex. Let's bring AI to bear on this stuff because it's going to be hard for human minds to disentangle these connections. But oh my God, what a future. The chemistry of biodiversity. The third theme, biodiversity baselines. And you know, it's pretty important that we understand how subjective ecoregions are as we begin to think about setting aside planetary spaces to preserve life. And really, we don't have a very good understanding except for some of the larger particles on our planet, the vertebrates. But for other organisms, we know very little. This is a painted map of the world that shows the ecoregions, and it looks beautifully delineated. We decided to ask the question, how subjective are those ecoregions? So we're using a standard collection method to look at the most diverse component of terrestrial life. There's the malaise traps, there's the bulk samples, and you extract DNA and you read the species in them. And doing that, you can very cheaply ascertain overlap among regions. We've done it across Canada. There are 15 ecoregions in Canada. Canada is a big place, definitely bigger than Belgium, a few thousand kilometers across. And I'm just looking here at the overlap in species composition in these 3 ecoregions. And if you look at the blue, the mountains out in Alberta, the red, the most southerly ecoregions in Canada, you can see their species overlap, and there's substantial species overlap, even though they're separated by thousands of kilometers. And in fact, if you look across Canada at large, it's 12% in these most distinctive ecoregions. And if we pool all the ecoregions, the figure goes up to about 30% on average. It's shades of gray, ecoregion in Canada. Then jump to Costa Rica. Go look at 3 ecoregions, rainforest, dry forest, cloud forest, that are just 30 kilometers apart. These are black and white. 3% overlap among those ecoregions where they're sitting basically on top of one another. This is the reality of biodiversity distribution. You need to know this if you're going to set aside protective areas. Parts of the world are gray, others are black and white. Where are the black and whites? Okay, so I mean, ecoregions are subjective, unquestionably. We need this information if we're going to develop well-strategized decisions in relation to land protection. Okay, where are we standing? Bioscan to planetary biodiversity. We set those goals in 2019. I shuddered, Torben. We had no money. Oh God. Anyway, we managed to achieve it. I mean, 20 million specimens. In fact, we're overachievers. 20 million specimens will be barcoded, so species discovery is advancing nicely. 2 million specimens will have their symbiome probed, and we're not going to exceed the target for the 100,000 bulk samples. It's a lot of work to collect all those samples from places all around the world and get the permits. Okay, we're poised for planetary biodiversity. Those are the big goals: register all species, reveal species interactions, and launch global biomonitoring. The original plan was to take a rest. 2028, settle back for about 3 years, raise the money. Giles, we need €1 billion. Canada will help. In any case, that was the plan, but we've confronted an uncomfortable truth. We now think there are about 24 million multicellular species on the planet. We didn't know the count was going to be that high. We can't afford to sit and take a holiday. We need to just carry on. We've been raising up the number of specimens, doubling the number of specimens that we've analyzed every 5 years through 2025. We weren't going to take a holiday largely from 25 to 30. No. We need to double and a half the number of specimens. We need to crank up the total to 30 million, and then we need to keep going. 60 million, 120, 240, 500 million specimens will give us 20x coverage for the estimated 24 million species of multicellular organisms on our planet. Okay. How do we do it? We're not going to take a holiday. Next January, we're launching a Linnaeus 2X. Linnaeus 2X has the goal of doubling the number of known animal species within a 5-year period. Take it up from 1.57 to 3.14 million, and to do it in just 5 years. And that'll set us on the target to achieve the goals of planetary biodiversity. What are we going to do? A lot of discovery work. Animal species register 3 million. Pete, we're not forgetting about plants. We really need to get into plants seriously, and we need to knock off the 500,000 species, but 100,000 is a nice number to add. We're going to do this through analytical nodes. We've got to move away from just core facility. We need to bring the world into this. And amazing new sequencing technology from Oxford Nanopore Technology slashed the cost of sequencers. But if you're going to use this in a distributed fashion, you need help. You need help with chemware. And so we created this package called Ontology, which provides freeze-dried PCR plates with all the UMI tags on them, DNA extraction, and the informatics that you need, because when you start cranking out 1 million sequences or 200 million sequences, you need some help, or many people need help to analyze the data. So that's what Ontology delivers. We hope by 2031 the world looks like this, and I'm sure it should. Core facilities generating 2 million, 2 nodes, 200,000 or 20K. And then, of course, looking forward a little bit further, the planetary biodiversity needs to turn this into, Dimitris, as you said, everyone needs to be able to do this. It needs to become so simple and create a mega network, and that's the way we will finish the registration of life on our planet and its protection and a deeper understanding of its interactions. So collect, sequence, analyze. There we are. And we won't do it without Europe. BGE is absolutely mission critical. You've got the scientific force, you've got the resources. We need help. This is a global problem. The global north is responsible for looking after life on the planet, working with our colleagues in the global south. And for the rest of the story, not 10 minutes, I've gone over. Anyway, 10th International Barcode of Life Conference next November in Bangkok. Please come and join us. Thank you.
Thank you very much, Paul, for that global vision. And now we follow up with the European perspective from Pete Hollingsworth.
Thanks very much, Paul. So yeah, my aim here is to then give the European perspective. So summarize the landscape, but also identify some key priorities. And the work of barcoding in Europe is supported by iBOL Europe, a community hub promoting connectivity, collaboration, and coordination. And when we look across Europe, we can see this very active, highly distributed community. Top right there, iBOL Europe, member organizations, but then this incredible heat map of barcode record generation. So a large distributed, highly productive community, albeit with heterogeneity across different countries and geographical regions. National nodes and programs are fundamental to success, and there are a range of different programs in different countries with varying levels of formality and resource bases. And just flagging Germany, Norway, and Finland as 3 examples of very well organized, well-resourced, highly productive national initiatives. But it's been really exciting recently to see the development of new barcoding nodes and programs, particularly in the south and east of Europe. The Bulgarian Barcode of Life, the rapidly expanding Polish Barcode of Life program, and fantastic to see great funding investment in Greece for the Greek Barcode of Life initiative. And these activities give rise to some fascinating foundational science. So comprehensive barcode reference libraries enable the production of practical resources like a molecular identification resource for the arthropods of Finland, but they also allow insights into large-scale ecological or evolutionary processes, such as this study looking at hybrid formation across the entire British flora. But moving from the national level, the production of data in a standardized fashion across Europe allows continental scale evaluation of biodiversity patterns, such as this analysis of European butterfly genetic diversity. And we're also seeing these Europe-wide perspectives, sharing practical expertise, practical experience, such as this very recently published paper looking at the role of citizen scientists in DNA-based biomonitoring. But of course, it's absolutely key that that science delivers for policy and meets the practical requirements of stakeholders. And again, It's activity at both national and European levels that are key for success. So the example here, one of the early examples of practical deployment of a DNA-based method for conservation in a regulatory setting is that of the detection of great crested newts in water bodies to support the assessment of development proposals. So that's operating at a very local level, and then right the way up to this wonderful DNA Aquanet program, which involves gap analyses of what was missing, but then also working right the way through to development of practical guides for practical deployment of methods. And these regional, Europe-wide initiatives complement very nicely the national-based activities. So there's now the relatively recently funded eDNA Aquaplan supporting eDNA work in aquatic systems across Europe. There's the Meta plant code. Birgit is somewhere in the audience. I've seen her somewhere waving there at the back. Again, wonderful project looking at harmonizing our approaches to plant monitoring with meta-barcoding. And the Global Spore Sampling Project, led by Finland and with great representation in Europe, using existing infrastructure for sampling air particles to provide new insights into the distribution of biodiversity. So building on this combination of national and regional successes, I just want to identify a few key priority areas. And as a segue into that, I've just put this heat map, which a colleague generated for me yesterday, on the distribution of plant species richness in Europe, just as a reminder that in all of our future priorities, there is a particular need for enhanced biodiversity characterization and biomonitoring in the south and east of Europe, where biodiversity is so high. So one of the first key priorities is that of curated comprehensive reference libraries. All DNA-based biomonitoring is only as good as the reference library that is being queried with those sequences. Curating the libraries, not just generating data, curating the libraries is key to make sure they're high quality. And I've just put here some tools from the Biodiversity Genomics Europe project, which are aimed at supporting the process of reference library curation. From the perspective of building those reference libraries in Europe, we have these fantastic, globally important biological repositories, and these are key to the development of reference libraries for Europe and wider global biodiversity. And it's fantastic to see so many developments in improving practical process in reliably and rapidly generating sequence data from these museum collections. And there's a strong priority to further develop these protocols to make it faster, cheaper, and more reliable to generate data, but also supporting this cultural transformation from a curator's perspective, that this is not taking something away from a specimen when you sample it, but you're adding to it, you're enriching that specimen with sequence data. The second priority is that of democratizing access to sequencing capacity. And in the future, we're clearly going to have the mixed economy of the big machine generating vast amounts of data. But it's key that that access to those technologies is improved. And as Paul mentioned earlier, the developments in the low-cost, portable, small-scale sequencers allow now the establishment of labs across a wide variety of resource settings and remove a lot of the barriers to participating in sequencing projects. And I think it's really important these approaches are rolled out, and we support their rollout in cases where there's high biodiversity but low income settings. And then the final priority I want to highlight is that of operationalizing our DNA-based biomonitoring approaches. Key to that will be standards, harmonized approaches, and you'll hear from Christian Meissner more about that tomorrow. And also the development of national strategies for building and formalize the process of integrating DNA into national monitoring programs. And Tina Lammerman is also going to speak about that tomorrow. I mean, we're at this absolutely key tipping point of transformative change in how we characterize and monitor nature. So we've got to validate and calibrate. We've got to make our methods backwards compatible, but not be anchored in the methods of the past. So we build on the past, but be very forward-looking. We're going to need to set new baselines. And in the setting of those new baselines, we've got to manage our samples and our data in a way that those baselines can be dynamic as new technologies evolve into the future, so that we really make the very best of this opportunity at national levels, European levels, and more widely to fully integrate DNA approaches into monitoring biodiversity. So that's me wrapping up now. So just the kind of headline take-home points. There's extensive efforts across Europe building reference libraries and delivering biomonitoring. Collaboration is absolutely key for the success of the endeavor at national and regional levels, flagging the particular importance in Europe of all of the biodiversity in the south and the east of Europe, where much biodiversity remains uncharacterized and needs to be characterized for its conservation and sustainable use. And then these three priority areas that I identified, everything depends on the quality of those reference libraries. We need them better quality and we need them better populated. Removing barriers to participation, particularly with low-cost sequencing technologies, and researchers and research users working together to enable this transformative change in how we use DNA to monitor biodiversity. Thank you.
Thank you very much, Pete. Okay, and now from the barcoding perspective, we're going to switch gear and move into the reference genomes perspective. And we heard from Paul for the global iBOL initiative, and we have the pleasure of hearing from Harris Lewin, who is the chair of the Earth Biogenome Project, who is in New York at the moment. So he will be talking to us through his video, which I know is 9 minutes, so this one will be on time.
Good afternoon to all of you there in Brussels. I didn't quite hear it, but thank you for the introduction, Rob. My apologies for not being there in person due to a scheduling conflict, but I'm certainly there with you in spirit. And I greatly appreciate all of your efforts as well as the efforts of all your colleagues in Biodiversity Genomics Europe. Today, I'm going to give you a brief overview of the progress we are making on scaling the production of reference quality genomes for the understanding of biodiversity. The Earth Biogenome Project is a confederated international network of networks that has the common goal of sequencing and annotating the genomes of approximately 1.6 million named eukaryotic species in 10 years. The EBP will proceed in 3 phases, with a sequencing of 10,000 species in phase 1, including a representative species of about 5,000 or half the eukaryotic families, and 5,000 other species of scientific interest or bioeconomic importance. Phase 2 will include the collection of 300,000 species and the sequencing of 150,000 species that represent now greater than 90% of the family's total and approximately 50% of the eukaryotic genera. Phase 3 will commence in 2032 after the completion of phase 2, and will include the sequencing of the remaining approximately 1.6 million named eukaryotic species. All of this, we have estimated, can be done at a total cost of $3.9 billion. which is $800 million less than our original 2018 estimates. And that is due to the improvements in DNA sequencing technology and cost efficiencies that have been gained across the entire sequencing and reference genome production pipeline. I might add at this point that 3.9 billion US dollars today is less than the cost of sequencing the human genome in 1993 dollars. That, adjusted to today's dollars, is about $6 billion. Obviously, at a cost that's about 2/3 of what the human genome costs. Sequencing of biodiversity is probably going to be one of the greatest values in the history of science. The Earth Biogenome Project is a confederated international network of networks and has an elected executive council from its membership and a committee structure that is responsible for the production of standards for the collection and processing of samples, for sequencing and assembly, for annotation, for analysis, and for information technology and informatics. There are also committees for ethics, legal, and social issues, as well as Committee for Justice, Equity, Diversity, and Inclusion. And this overall structure is mirrored in the ERGA structure that has been created for Biodiversity Genomics Europe. At present, there are 60 affiliated projects with more than 2,000 participants located in 88 countries around the world. All of the projects are committed to an open data access model, compliance with the Convention on Biological Diversity and the Nagoya Protocols on access and benefit sharing, and the use of digital sequence information. The Earth Biogenome Project is also committed to diversity, equity, and inclusion within the EBP community of scientists and engagement with indigenous peoples and local communities. The Global Secretariat for the Earth Biogenome Project is located at Arizona State University, with additional support provided by the University of British Columbia and the Wellcome Sanger Institute. As my colleague Mark Blackser, who is in the room with you, likes to say, sequence locally and think globally. As just mentioned, the Earth Biogenome Project is a network of networks. This global network is being nucleated around nodes that coordinate sequencing across a broad geographical area, such as the European continent. In fact, ERGA, the European Reference Genome Alliance, is the first regional node of the Earth Biogenome Project, and some of the major projects and contributors to the EBP are organized under the ERGA regional node, including the Darwin Tree of Life project located at the Wellcome Sanger Institute, the Atlas C project, and the Catalan Initiative for the Earth Biogenome Project, all major contributors to the production of reference quality genomes at global scale. So what is the current status of the Earth Biogenome Project? The EBP has now passed a major milestone of producing more than 4,300 genomes. That represents about 20% of the total number of genomes in public domain databases. If you look at that as a representative of the number of genera, that's about 1/3 of the number of genera and about 44% of the number of genomes for each eukaryotic family. If you look at year-over-year scaling and quality, the EBP affiliated projects have been doubling the number of genomes produced every 2 years. And if you look at the light blue shaded bars, you can see that the largest increase has been in the number of chromosome scale assemblies. The numbers for 2025 only represent the first 8 months, and we fully expect to hit close to 5,000 genomes, about the halfway mark toward phase 1 goal by the end of the year. To reach the phase 2 target, we're going to have to scale more than 10-fold the current output globally for genomes. That is a major challenge that the project faces over the next year or two, to understand the requirements and to develop all of the procedures and protocols and obtain the funding to reach the phase 2 goal. One of the most impressive aspects of the Earth Biogenome Project is the fraction of reference quality genomes produced by EBP-affiliated projects and the rest of the world. If you look at the data at the species level, EBP-affiliated projects have produced more than half of the total of reference quality genomes. At the genus level, it's 60%, and at the family level, it's 70%. Nearly 70% of all reference quality genomes, and we define those as those genomes that would meet the minimum EBP standard metrics, as shown at the bottom of the slide. So I just want to review and finalize my talk by going over a few key points. First, reference quality genomes can be produced at scale. We have demonstrated that now over the past 7 years. And that will enable us to achieve our goal of sequencing all named eukaryotic life in a 10-year time frame. The EBP is an international network of networks that has taken years to build this network. It is now highly integrated and very successful in terms of producing the standards that are necessary for the project and facilitating global interaction and collaboration. And to achieve the goals of phase 2, the regional nodes and the affiliated projects must work together to scale all phases of the production pipeline. And that is going to be the main focus of our activities over the next few years. And as mentioned previously, ERGA is the first EBP regional node that is coordinating production of reference quality genomes on a continental scale across Europe. And ERGA will play a key role in providing solutions for the biodiversity crisis, enhancing the resilience of agriculturally important species, and stimulating a circular bioeconomy. The EBP looks forward to continued leadership of ERGA and of Europe, and I wish you success with all your efforts over the next few days. Thank you very much, and now I'll hand the floor to my colleague Camila Mazzoni.
And Mark, could you just wave? Where's Mark? There, okay. So Mark is Harris's de facto deputy. So if you want to speak about EBP, go and find Mark. Now we will move to the European perspective and hear from Camila Mazzoni.
Thank you, Rob, and thank you, Harris. So as Harris said, ERGA is the first European or first regional node for the Earth Biogenome Project. Just showing again the map that Harris showed. Here in the black dots, you have all the affiliated projects for EBP. And in Europe, we have ERGA as the central yellow with many new dots or many new connections that actually connect EBP to countries and initiatives that are not directly connected to EBP. So the role of the regional node is much bigger even than coordinating all the projects at the continental level, but actually activating new projects and new countries that are not yet part of the EBP network of networks. The way that ERGA does that is by inclusivity. So we have the council where we have representation from 38 countries across Europe with 1 or 2 representatives for each of the countries. We built the ERGA structure bottom up, now trying to reach this inclusivity with this structure here that you see in a sort of half a network where we have the central structure from ERGA consisting of the council, the executive board, and committees. The committees mirror those of EBP that Harris already mentioned, and these are the ones that allow us to cover all the aspects of the genome generation on the 5 committees on the top that goes from sampling to sequencing to assembly and annotation, the data analysis of these genomes, and all the way to the infrastructure and data handling. But building genomes is much more than science. It's actually, in order to make them useful for society, we have to connect and communicate and include society. So here we have all the ethical side, the communications, citizen science participation, training and knowledge transfer, so our capacity building aspects, and the inclusivity in the Social Justice Committee, all contemplated in these 5 Science Plus committees. Our committees are open, which means that they serve as a knowledge hub for each step of reference genome generation and use. So that's where Europe is invited to join and discuss together about these aspects and develop and evolve together. But very importantly, we have the affiliated projects. As the EBP regional node, we have a role of joining these initiatives, big and small, under our umbrella, so that together Europe can sequence a high amount or even accelerating amount of genomes. So to this date, under the umbrella of ERGA, we have a number of projects, including some of the major ones, such as Darwin Tree of Life, the Catalan Initiative for the EBP, which Harris mentioned, and Atlas C, which is a French initiative that actually got support from ERGA to get started. In total, we have 2,650 species now under the umbrella of ERGA. Apart from these affiliated projects, ERGA itself has organized across these different countries in order to activate the genome production across Europe in countries with more and less infrastructure. In our pilot, which is now published in NPJ Biodiversity, we were able to sequence 95 eukaryotic species from many different countries, at least one per country, putting together here 24 academic sequencing centers, plus 3 other laboratories and a museum biobank, counting with support from 9 worldwide biotechnology companies in order to, without any budget actually, put this pilot together and activate all the countries that you see here in order to have, for in some cases, the first reference genome sequence for that country. Apart from that, ERGA has been producing genomes as well as part of the genome stream of the Biodiversity Genomics Europe. And in this case here, we had a smaller amount of sequencing centers, but it was much more important here to harmonize the genome production across Europe. With 7 sequencing centers in 6 different countries, we managed to make them work together in a workflow that is highly harmonized in the way that we produce data, that we handle the data, and that we make this data available. With this, we are going, by the end of the project, to sequence about 290 karyotic genomes from many different countries in Europe, spanning 480 gigabases of genome size. So with this, ERGA is not only this knowledge hub that I mentioned to you, it's also the umbrella network for all the projects producing reference genomes from European biodiversity, the big ones and the small ones as well. And apart from that, we produce reference genomes, but not for the sake of producing at a scale from within ERGA, but for the sake of learning how to work together and how to work in a coordinated fashion across a continent that can be very different in terms of infrastructure and culture as well. And beyond that, we have all the connections with society, with stakeholders, with research institutions, and many associated partners that either are in Europe not producing genomes, but maybe using these genomes, or are outside of Europe producing genomes for other parts of the globe. And with this, then ERGA becomes a community-based coordination of reference genomes generation and use in Europe. For the use and for the use of genomes and bringing these genomes into real application, we have among our members, more than 1,000 members across Europe, the focus research of many different types of taxa and many different types of applications. In the BGE genome stream, we had 29 case studies under the flags of bioeconomy, conservation, and One Health in different countries across Europe, showing how reference genome and high-quality genomic data can be used in real applications. Apart from that, it's very important that we connect beyond UGA, beyond our own consortium, and the stakeholder articulation is something that is already really important for us and is becoming more and more something that we're going to pursue in the coming years at the global, European, and local scale. So initiatives such as GeoBond and GGBN, Genoa, and also EBOC, which is going to get started sometime soon. We aim to be part of these bigger initiatives and support the production and, more importantly, the correct use of genomic information. Apart from that, all these case studies, the ones that we fund and the ones from our members, they support and connect to hundreds to thousands of stakeholders all across Europe at the local level. With this, how we see the perspectives for the future of ERGA and the uptake of genomes in biodiversity policy, we need this largely interconnected network of genome hubs, truly interconnected, working truly in synergy. And this way, we're going to be able to scale up the production and also the use of genomes. So with this, we say that ERGA, or biodiversity, needs to be understood, to be protected, and science needs genomes in order to understand biodiversity. This is the level of knowledge that has been missing for us, but now we are capable of generating it and at scale. Thank you.
Thank you very much, Camila. Okay, so now we've heard from the barcoding community and from the reference genomes community, both at the global level and at the European level. And now we come full circle back to have Dimitris present the progress and updates from the Biodiversity Genomics Europe project itself.
Thank you. I think it's great that we heard from Paul and Harris and Camila and Pete and Rob about all the amazing things that are happening at the global scale, that North Star that we have, these mission statements, initiative that we have seen, and we are working in the European counterparts of them. And of course, as I said in my introductory comments, all of that needs to always come in the context of the urgency and the need for acceleration. I mean, Camila, you finished saying we need to accelerate, and that's exactly what I'm trying to say as well. So one thing is for sure, that we're not embracing the future fast enough. And if we really want to achieve that level of impact across Europe, we need to do business in a slightly different way. Paul, Harris, and all of us, we're talking about mission statements. We're talking about missions that connect us, that allow us to share the way we look at the future. And if we really want to achieve this kind of missions, we really need to improve two things. We need to improve our efficiency, how we are able to deliver more with the same resources. We really need to improve the effectiveness in which we're doing business. How we reduce effort replication, we share resources and expertise, we orchestrate, coordinate our effort, and how we deliver, for instance, openly data and knowledge so that we can allow others to benefit from that. How we continually build capacity and engage our existing assets that we have either locally or shared assets, and establish more of a service-oriented mentality that allow others to onboard and benefit. So if there is something I'm very proud about BGE, is the fact that as a European initiative, it managed to bring together these two communities. And managed to bring together these two communities not because it was easy, but because it was necessary. It was absolutely necessary if we really want to improve the way we do business, that these two communities start working together at a continental scale. So iBOL Europe and ERGA join forces together with a wider community of stakeholders, for instance, within our taxonomic community, to be able to deliver more. We brought together this proposal, started back, as I think you've heard, in 2022. The project has 32 beneficiaries, 20 countries. It's ending in the next few months. There's a lot of things to deliver. But it had two fundamental goals. One was to really, a scientific one, how we can accelerate the use of genomic science, how we can provide and enhance our understanding of biodiversity, how we really, by bringing together these two communities, by bringing together the people that are working in all of these domains, we can really show value, how we can create these economies of scope around them. The other part is more of a systemic change. It's about how we allow our work, how we allow our facilities, how we allow our expertise, our people, our data to connect. How we allow data to flow between different organizations, how we allow samples to flow between different facilities, how we allow expertise to be shared. And the way we decided to do that is to be able to pilot all of this within the single project, starting from increasing, supporting and enhancing the capacity of our nodes, whether it is the ERGA nodes or the iBOL nodes, showing how production can be achieved in a transnational fashion, going beyond boundaries, geographical boundaries, beyond the limitations that you would typically have when you are embarking on a project on a local scale, and show the value of all of that. And we wanted to do this because there is a plan behind it. And the plan is how we are going to be able to scale all of this. We want to show that this is not only necessary, but it is possible. It is feasible. The only thing that requires is good orchestration and coordination. And we need to make sure that we take into consideration when we're talking about the investments we want to make in achieving these missions, we need to take into account the European reality. And the European reality is slightly different. It really is predicated on the requirements of national governments, member states, to invest in these mission statements, in these missions and the goals. It's predicated on the distributed effort by default. In Europe, it's very difficult to get 100 billion or 1 billion or 4 billion euros to invest from a single source. But this money is already available. It's already available through the national efforts, through the national investments. If only we can connect it. If only we can align it so that it delivers value. So that's what we did. And the structure of the project was following exactly that. So you see this in different streams, the horizontal perspectives, essentially the barcoding stream, a genome stream in red, and a consolidated effort stream, which actually showed value from the collaboration of the two communities. And then the pillars, the columns here, basically represent, apart from the first one, which was node development, capacity building, represented an operational value chain, essentially coming from sampling to sequencing to data to application. So you can see here that there is a very clear link, a dependency between all the different work packages. And behind all of these work packages were different organizations in different countries relying on each other to deliver the end product. And this is extremely difficult to do in projects like this, where you really need to create these operational dependencies and pipelines and maintain them throughout the project. But it was possible. And that's exactly what we wanted to show. I'm going to breeze through some of the, because I think it's very important to show also some of the outputs of the project. Whether it is, and I'm taking it from the stream level, so whether it is in barcoding, Pete alluded to some of these developments, that we managed to have through the capacity pillar in barcoding. In sampling, you can see some of the, not huge numbers, but remember that the idea here was not really to create a lot of content, it was to showcase the feasibility of the model at scale. And of course, in barcoding applications. And similarly, on the genome front, with a lot of developments also on the node capacity, for instance, with the ERGA Knowledge Hub that brings together all of that generated information across the different projects. In the production, and in the application. A lot of attention was put also on the joint stream, how the two communities start to share things together, share practices, knowledge, understand the possibilities for shared components of the future. And this is exactly what the join stream was all about. And we took different application areas or case studies to see how this can happen. Whether it is in dark taxa or in genomic barcodes, identifying new barcodes or data management or specific groups that we would like to focus on, and of course the training, which also was a very important aspect on how we went about the project. Policy and ethics were equally important. Navigating the transnational sample management in an ABS world is not that easy as well. So many of the things we found as pain points in the whole project, apart from this maybe that we expected it to be an issue, were issues that we didn't even think about. How you manage the DHL shipment from one country to another. I mean, we designed a project that was very aspirational, but we designed an airplane and we stumbled in building the terminal. I mean, but these were lessons that we learned. And this was exactly the things we wanted to achieve. The lessons we learned through this project are even more important than the successes it had in content. And we want to capture many of those lessons in some policy briefs that we hope that they will help us, but help the rest of the community move forward. As I said, BGE goes beyond the boundaries of a project. I believe that actually it forms an initiative, bringing together the communities. We were happy that back in 2024, was an MOU signed between ERGA and iBOL Europe to substantiate that kind of commitment to a joint future. Looking beyond BGE as a project, there's a lot of things we would like to do. And of course, funding is always necessary. So we didn't miss the deadline, Gilles. But one of the few things we want to see is how we can start supporting and orchestrating this distributed data production. How we can move beyond the boundaries of a project into basically a community-supported mechanism for knowledge production. Investing in innovation and scaled-up biodiversity genomics, focusing where innovation is really needed to reduce the friction in that transnational collaboration. Work towards that organization of the community in such a way that allows us to orchestrate better the work, to inform decisions about what you need to do and what has been already done. And of course, we need to do this by continuing to strengthen the networks and the nodes that basically are the heart and the soul of what we do in biodiversity genomics. So I think there is a brilliant future ahead of us, but it requires the support and commitment of all the actors in Europe. Thanks.
Right, thank you very much, Dimitris. So we do have now 20-ish minutes for Q&A and discussion here at the end. For those of you who are deeply involved in the Biodiversity Genomics Europe project and in either the ERGA community or the iBOL Europe community, a lot of what you've just heard is probably quite familiar. But this first session was really for all of our invitees here, right? To bring some context, understand who we are, where we're coming from, both from the perspective of the community levels, the global levels, and then also how this ties into the Biodiversity Genomics Europe project. So we can now open it up to the floor, and I will try to give priority to non-BGE audience members. And I can try to get a microphone to you. Who's brave enough to go first?
Yeah, no, Amber Hartmann-Schultz from the Leibniz Institute DSMZ, which is based in Germany. So I'm a microbiologist, maybe a bit of a freak in the room relative to all you metabiologists, or whatever the opposite of micro is, but maybe a provocative question. I think these global efforts are, of course, incredibly powerful, help to meet broad range planetary ambitions like the Global Biodiversity Framework, but yet I can't help be struck that you've forgotten some major actors in these. And I'm not forgotten, I know that there's reasons why that is, but I would love to hear thoughts on when and how and if and whether the major drivers of the planet, which arguably probably are the invisible things, like when do they come to the table of biodiversity genomics? Because I had the chance, sorry, this is now less of a question and more of a comment, I had the chance to participate in the founding of the first International Union for the Conservation of Nature, so ICN's microbial specialist group for conservation. And the papers that we've written in the last 6 months have really made this call like, hey, we've been forgotten for a long time. Not we microbiologists, but the microbes on the planet. So any high-level reactions of when those, not only barcoding and full-scale genomics meet, but also when the microbes meet the rest?
Maybe I can pass this one to Paul first, because your slides had the most with things without a name yet.
Where am I going to get a mic? There's the mic. You're absolutely correct. But, you've seen the budget numbers that we're talking about, just trying to focus on multicellular life. We're going to need to raise more money than have ever been raised for biodiversity genomics research. So that's the challenge. You don't want, and I think the history of science has shown that it's a really good, if you take a look at the big sciences like astronomy and particle physics, they schedule their projects, they tackle things that are achievable, knowing that there's a lot more out there. And, we save every DNA extract and their whole body extracts. And we know they're filled up with protease, bacteria, and, nothing will make me happier, viruses, than the day when we can start to look at that material. And of course, you can't avoid looking at bacteria any longer. When you're doing high throughput sequencing, guess what pops up? Bacteria. And that is, the connections of life is just where I see this going, and we can't ignore all of the organisms that fascinate you. So we're ready. Please find the additional money.
Thank you, Paul. I think there's a hand up there from Mark.
Here you go.
There's one coming to you from the left. Sorry.
Just to comment on this one. Those of us doing eukaryotic genomics have always bump into the prokaryotes, and we assemble them and we release them as prokaryotes associated with the host. So, for example, Tree of Life at the Sanger Institute has sequenced about 3,500 eukaryotic genomes. We've actually released about 12,000 prokaryotes. So that's the host-associated microbiome, the cobiome we call it, which are pathogens, symbionts, gut bacteria. There are efforts afoot to sequence the world's prokaryotes, usually from metagenomic samples rather than isolated specimens. And those are now at the levels of several hundreds of thousands of assembled genomes. So I think that is actually the way forward for getting at that set of hidden diversity. and also the vast hidden biochemistry that these things can do. So, for example, there's a Danish project which is a microbiome analysis, a microbiome analysis for the globe. And they're sequencing from soils initially across Europe, but aiming to do a bioscan-like thing, but with deep, long-read metagenomics across the world. So I think it's coming. It's coming in a very different way because we can mostly isolate individual specimens and sequence from that specimen, whereas for the bacteria it's having to be done as part of consortia and communities and ecosystems.
Thanks for adding that, Mark. Kasia, was that a hand or was that a no?
No, I'm
okay.
speaking in the mic.
Yes, if we can get a microphone down to Camila, thank you.
Yeah. So I wanted to comment on this because when we think about monitoring or the applications of these technologies, I think the microbial world is actually way more advanced than we are in terms of looking into soil health or water contamination. So the metagenomics is actually quite advanced in already applying genomics. The problem at what we have, the eukaryotic level, is this huge complexity that we have to deal with. So it is true that BGE, iBOL, EBP are focused on eukaryotic biodiversity. But at the same way that we are not going to cover maybe the entire prokaryotic world, we are also not going to cover all types of biodiversity monitoring using genomics, and we are not even going to try to do this. I think in the future, what we're going to see is that genomics is going to have its place sometimes as the central technology, sometimes as complementary, sometimes just there to maybe create a baseline for other technologies that might be quicker in that very specific task. And I think the prokaryotic world and the eukaryotic world will meet each other because it's unavoidable, but I don't see at least a big focus on the prokaryotic world directly coming from BGE. Having said that, in BGE Plus, one of our major goals, and we have a whole work package for that if we get funded, this is going to be completely focused on establishing all sorts of connections that are needed in order to integrate genomics from the eukaryotic point of view with all other types of technologies, including there the prokaryotic genomics analysis. Yeah.
Thank you. Here in the front.
Hello, I'm David Eggers from the Netherlands, from Waardenburg Ecology. So in this discussion, I kind of represent the bioeconomy side. We are an ecological consultancy company. And I was wondering, I hear a lot of talk about like increasing, standardizing, making the databases more complete. But I was wondering, I don't hear that much about the application side. So how are we gonna, I look at this post, it says Europe's drive to reverse biodiversity loss through genomics research. I don't hear much yet about like how do we actually use this genomics data in practice to achieve this. Can somebody comment on this?
Are you gonna stay for after the coffee break?
Yes, for sure.
Okay, there we go. Is the very short answer. So yes, we focused on the context for now, and then we're going to move into the applications from the barcoding perspective and the genomes perspective after the coffee break, with many examples from the BG, but also from our invited speakers here today. There was another hand here somewhere. Yep.
I just would like to perhaps address the question about the microbes. I think besides all these efforts to produce the data at scale, we should not neglect technology development and also development of the protocols that BGE is doing, because for eukaryotic microbes, like for microscopic fungi and for prokaryotes and for diatoms and for everything else, there is a huge gap in technology of how to actually sequence reference genomes, for instance. And here, I think the BGE and the Earth Biogenome, not just setting standards, but also trying and pushing the existing technologies more and more to the uncharted territory. How can we actually map that hidden biodiversity in the eukaryotic world as well? So need more money for R&D.
More money for R&D, yes. Any other questions? Yes, go ahead, Gilles.
As we speak about funding, and I was invited, we have some euro millions, but we do not have euro billions. And actually, there is a big role for the private sector, and they have money. And actually, when we had the agreement on the Kunming-Montréal Global Biodiversity Framework, you had the big DSI and ABS angle. And probably some parties, they agreed because they wanted that. So in Europe, we have still leadership, but we need to work with the private sector. So they have also to fund. In terms of EU money, so it's not only the specific topics, as I said, we are now really working to mainstream biodiversity in many different angles under Horizon Europe. And so please have a look when you will see the work program 26. The info days are already decided. Caroline, correct me if I'm wrong, but I think it's 22, 23 of January. And also when you look at the partnerships, so Biodiversa Plus was mentioned, but there are other partnerships working with or could benefit from DNA. So keep that in mind. It's not only Europe, it's also, as Dimitris said, national fundings, and this is where it's very, very important to use for example, the connections thanks to Biodiversa Plus, but not only. The good news is that the member states signed an MOU this year about infrastructure, and we are talking about 600 millions, not necessarily for biodiversity, but it means that if all of you go nationally to your contacts, you can help get more money, national funding. Two last things, and I'm losing my voice, excuse me. At the international level, you have now the launch of the TSC, so the Technical Scientific Coordination Center for the CBD Secretariat. One of them for Europe was officially launched last week here, and it's coordinated by the GRC. So have a look, because this is really where you can find international collaboration. And last, regarding IBES, we will receive the very first draft of the monitoring assessment in November. And I have to check what is the deadline to take into account publications, and particularly the use of genomics for monitoring. But there is still time, so please publish and alert us so we can use European research. Thanks.
Thank you very much, Gilles. Good sound advice there. Any other questions from the floor, from what we've heard of so far? Then just to bounce back off Gilles there, so what I didn't say in my introduction is that I'm also the chairperson of the European Reference Genome Atlas, and therefore in our capacity as sort of community guidance, we are really encouraging diversification. So at national level, at regional level, within your own universities, etc. You know, thinking small but in a connected way. And I think in that role we are trying to play is to give people the confidence to be able to put forward ideas, to put forward projects, even if they themselves have not yet been successfully managing and running projects in full-scale genomics. But because of the support of the community around them, they have the confidence to at least have these conversations nationally, locally, with their institutions, etc., to advance the cause, if you like, to eventually translate into something much, much bigger. Any other questions or comments from the floor?
We have an online question from Barbara Salih. It's one about the communities. What are the most effective outreach efforts today to involve more countries in more intensive participation?
So I would hand that over to Camila and then Pete. Did everyone hear that? Yeah. What are the most effective ways to engage? Yeah.
I wish I had an answer. we try many different things. We try to be inclusive because, I mean, at ERGA, we are looking at individuals first. Our members are first individuals, then the institutions that come with them. So we empower individuals. And what does happen is that many individuals come maybe feeling that they are not capable of being in the same discussion as others that have been there before. And I think this is, we have to make room and make them feel comfortable and make it sure that they understand that they're welcome there. And these individuals are the ones that bring their countries together. These individuals are the ones that bring the institutions. And I think this has been our approach, but we change, you know, because Europe is so complex and big in terms of countries. I mean, thinking about handling 38 different countries, we have to be always flexible in how we approach and always open and inclusive. And that's why we just created recently a new committee for inclusivity, social justice, to make it sure that we are bringing all of ERGA, all of Europe, together with us. But there is no magic formula, I'm afraid, or at least if there is, we don't know it. Do you, Pete?
No. Yeah, I mean, it's a really good question. And actually, within the iBOL Europe community, we had a workshop where we got together and said, what works for you? What works in different settings? And not surprisingly, there are some commonalities, but there are also lots of different environments that people are operating in. And one of the things that really jumped out is for a community that is incredibly collaborative, what we didn't tend to do was, when we set up national nodes, is ask people what worked for you. So there was a kind of really great realization moment. We should talk more about what were the best approaches for speaking to your funders, what were the best approaches for getting that national scale impact. But actually, I'm going to try and give a more detailed answer to this question. I'm just going to hand the microphone to Alexandros and Michel, if they don't mind, and I'm going to give them 2 seconds notice and keep talking just while they have a little thought about this. But for both Greece and Poland, there are 2 examples of countries which have really accelerated the scale of national activities. And I wonder if you both might reflect on anything that was key to making a difference, key to success.
Okay, so Alex, you go first. Yeah. Oh yeah, okay. Yeah, so if I need to go first, well, I think it all starts with passion for nature. That's one thing. And then it's a lot about social contacts and involving different people depending on your country. Like for us, it worked really well to talk to people from all these nature conservation agencies, national parks, landscape parks, and so on. And eventually right now, we are about to submit a large project with 40 landscape and national parks in Poland to do the malaise traps campaign, and also a big thing, this DNA of music, you know, thing to make people more sensible about the sound landscape of nature, and somehow to translate the science into culture. And you know, these people from the stakeholders from the national parks, landscape parks, they're extremely enthusiastic about that. But also another thing that must be very clear from the very beginning is that you must be very fair. Yeah, so you just need to offer them something and be honest about that, and not just make them feeling that they are somehow used by the scientific community. And this also is the same for the citizen scientists and so on. And that was, I think, yes, indeed, as Pete, you nicely said that POLBOL is rapidly expanding, and it is. I think that's true, but it's because of what I just said. So, Alex?
And I will quickly add as well that what was most important even in Greece and beyond is that we are open to collaboration and we disseminated as much as we could. We invited people and we were open about what we did. It started globally from Canada, but then we put it like a community building exercise. And what we're doing and what will soon be out as well is a paper submitted and shared with the community about what are the good practices and what are the challenges and what are the ways to solve them. So we're always here and we continue to be here building this community and telling people that the democratization of the technology makes it people more easier for them to trust, because now it's not like you're going to send your samples to that country and then they will come back to you. So this also helps a lot.
And just at the end, I think that this Polish-Greek collaboration is very fruitful. Also because, we are in different parts of Europe and we can learn from each other a lot. And it's an excellent friendship being born.
Was that a hand up at the back there? Yes, thank you.
Thank you, Birgit Gemeinholzer from Germany. I have a question. Is there a chance that there might be a possibility to develop a biodiversity offsetting similar to the emission offsetting and to finance these really huge projects which are so urgent?
Who wants to answer that one from our panel of speakers today? I think it's you, Dimitris. There's a microphone coming here.
Was it, just to confirm, your question is whether we can leverage biodiversity credit? Was it, you said credits? Yeah, okay. Obviously, yeah. Well, before I think we can learn first of all with what's happening in the UK. Obviously, the biodiversity credits discussion now is taking off also within the Union. But the UK has moved forward in implementing a nice scheme about this. Yes, but the connection is not so direct. I think what we need to understand is if we're able to go down the path towards a biodiversity credits scheme that makes sense. Right now, most of these biodiversity schemes, credit schemes, are relying on land use and change of land use. But obviously, this is a developing discussion, and we need to understand how our ability to provide more granular information about the state and change of biodiversity will be plugging into that kind of frameworks. And this is an ongoing discussion. At the end of the day, it is very important that we end up in mechanisms that are reliable, feasible, and simple enough to be implemented. And if you take the idea, because I've been having this discussion for some time now, starting from the carbon credits, obviously when they started, they were like very generic implementations. They grew, they developed, they're not perfect still, but they established a mentality around how we connect these natural assets to the bottom lines of organizations and companies. I also see more opportunities for financing coming from what I say, either the compliance, we see the needs for compliance from companies in terms of policy frameworks like the CSRD or globally in TNFD framework, and from what I usually say, the business risk analysis. We see more and more organizations really looking. We had a very big conversation a few months ago with ECB about how ECB is trying to implement the concept of financial risk profile connected to biodiversity, the way it's already connected to climate. Because it's very important for banks to be able to assess their exposure to risk, business risk, related to how those companies are relying on natural resources. So that double materiality of a company with a natural environment, impact and dependency, becomes more and more important in assessing their financial future, and as such, it is reflected in their bottom lines. Because more risk means more expensive. And that is something we would like to explore more, because there we can really see opportunities through which, through the investment of the companies, we can actually produce more data so that we can become more granular and more focused on the advice we give about the impact that every economic activity might have on the local biodiversity. That could roll out to a scheme of credits or connected to credits, biodiversity credits. But for me, the first step and more direct step is the one I explained.
Thank you very much, Dimitris. On that note, we've now reached the end of this first session. So you've seen the names and faces of some of the leadership here today, and we will be having a short coffee break of about half an hour. Anna or Gabriella, do you want to explain where it is? Because I see Arbin's VIP room, but I don't know where that is myself, not being from here. Thank you.
Yes, thanks, Rob. Indeed, our coffee breaks and also the lunch tomorrow are in the VIP room. If you just leave the room through that exit, you will find signage which says coffee, breaks, and lunch. So just follow them. It's about 2 minutes walking. So please enjoy your coffee, and we will meet back here in half an hour. Thank you.
And thanks to all the speakers. Thank you very much.